let's put this together. We've talked about the social brain, we've talked about large-scale alterations, and we've talked about genes for schizophrenia. How did the human brain evolve to be so social? Well, by the scenario I've been telling you about this evening, we've had a series of genetic changes, mutations, all along here, involving these highly developed traits in humans, and these were selected for. We've had the evolution of alleles for more complex sociality, and so the social brain has, by this argument, evolved in part via selection of alleles for schizophrenia. And if this is true, then schizophrenia is due in part to certain individuals having essentially too many of these schizophrenia risk alleles. Okay, so we've all got a certain number of schizophrenia risk alleles as underlying our social brain, but if you get too many of these, the social brain is overdeveloped in the sorts of ways that I showed you, and it's in the overdevelopment of these sorts of social traits. Now, the bishop thinks we have gone way too far and that he has us at last, because he says, and you may be thinking, justifiably, that alleles for madness are bad. Madness is bad. Alleles for badness must be bad. And such bad alleles, by definition, could not possibly have increased in frequency under Darwinian natural selection. So how are we going to answer the bishop now? I tried to do so a couple of years ago, working with Kyle Summers and Steve Doris, by taking a set of genes that have been relatively well linked to schizophrenia and looking for evidence of positive selection of those genes using data across mammals, genetic variation among mammals, and variation among human populations. One can search for these signatures, these genetic signatures of selection. And we found such signals of selection for the human lineage for roughly a third of these genes for schizophrenia, which was a significant enrichment in signals of selection compared to a set of 300 control genes which are engaged in neuronal activities. Now, I need to be very clear on what this means. This doesn't mean that this is evidence of selection for risk alleles for schizophrenia. This just means that these genes there is evidence that these genes have been subject to selection. The genetic variants that have been looked at in terms of schizophrenia tend to be different from the genetic variants that have been looked at that you can use to test for selection. So we can't really say if this positive selection, for the bulk of these genes, we can't really say if this positive selection has been selection for alleles which are protective against schizophrenia or alleles that are actually schizophrenia risk alleles that have been selected for it. Or presumably there could be some combination of those two. But we do have such evidence for specific genes, some of which I will show you. This G72 gene is one of the best replicated genes where you have alleles affecting risk of schizophrenia. It's involved in synaptic function, these little Pac-Men here. And it's possible to score the genotypes of individuals in normal populations like us for the alleles in of this gene, and some of those alleles are the risk alleles for schizophrenia. Okay, so some of us have those. These are relatively high frequency common. Now, if you've got one of those schizophrenia risk alleles, if you have the high risk allele for the G72 gene over here, this is not schizophrenics, this is normal people, then if you have this high risk allele, you have better verbal working memory, essentially better verbal skills for this component of verbal skills, significantly better than individuals who are at relatively low risk, who have the relatively low schizophrenia risk alleles. So So in this case, the schizophrenia risk allele in normal populations looks pretty good. Comparable patterns as these have been found for a couple of other genes that have been strongly linked to schizophrenia, and further work along these lines will no doubt be forthcoming. One of the most famous social brain alleles in humans is the FOXP2 gene. This is a gene for speech and language. It was discovered because its effects are segregating in this large family in London called the KE family, and roughly half of these individuals have this loss of function effect such that they have considerable difficulties with articulate speech. So this is basically the closest that we have to a gene for language, this critically important social brain function of humans. We have good evidence for adaptive amino acid evolution just in the human lineage for this gene. We have had 2 changes in amino acid in this gene in the human lineage, and both of the changes have involved amino acid substitutions that have effects on function. And they've put the human FOXP2 gene into mice and looked at the effects there. So the gene has clearly undergone adaptive evolution in the human lineage, and there's a specific polymorphism in this gene with this lovely name here that has been linked both to schizophrenia risk. So schizophrenia risk involves a lower percent cytosine, lower frequency of cytosine at this particular locus, at this particular nucleotide site, and autism risk involves the exact opposite, a higher percent of cytosine. So here we have evidence of selection and connections to both autism and schizophrenia, and wouldn't we like to know what the effects are of variation in the cytosine versus the other alleles of this gene? We do not know anything about that as yet. So we have just developed a conceptual model of the evolutionary basis of major human mental illnesses. We've talked about these highly developed in human traits here, how they are specifically underdeveloped in autism and specifically overdeveloped in schizophrenia, bipolar, and depression. And we've talked about how we have this sort of a continuum in social brain development. So we have a model, we've got a scenario, we've got some ideas, and we really need to test it, find ways of saying, well, is this really true? Do social brain genes underlie this continuum, and have they been involved in the evolution of the human social brain? So we've taken an evolutionary perspective, looked at genomic changes at many loci, changes in gene expression mediate the evolution of the large human social brain. We can also take a developmental perspective, think about these genes underlying social brain development in terms of being expressed in individuals at either lower or higher levels. So to try to evaluate this model, we want to look for relatively large genetic or gene expression changes where changes at the same locus will cause either underdevelopment in one direction or overdevelopment in the other direction in aspects of the social brain. Formally, this is essentially the same as those genes that I showed you that were involved on one hand in microcephaly and on the other hand in macrocephaly as a result of changes in expression that go in 2 diametric directions, except here, instead of just brain size, we're talking about complex social brain functions. So we want to look at large-scale changes because then we can really see the phenotype and quantify the phenotype much more clearly.